Hello, and welcome to yet another lecture on muscle tissue, and in particular skeletal muscle tissue. Today, we're going to be talking about adjusting tension in a whole muscle. So let's write that down as the first thing in our notes. So, adjusting tension in a whole muscle. And what we mean by this is how are we going to produce a contraction which is commensurate with the amount of work that we need to do, and that is not a jerky movement. So, how, and the way we can express that is how can a muscle contract in a graded manner? In other words, with the right amount of force for the job. And number two, in a smooth, non-jerky fashion. So the difference between picking up this pen and a heavy box. I'm going to use the same muscles to pick up this pen and a heavy box. Both of those require different amounts of force. How does that happen? How do we adjust that amount of force in order to accomplish the job that we have in mind? Furthermore, when we do make a movement, we want a smooth movement. We want to be able to move our hand in a smooth movement like this, not a jerky movement. How does that happen? Because keeping in mind the contraction cycle that we've talked about is a all or nothing process. In other words, it's all on or all off. There's nothing in between. So how are we going to get these two ideas met? Okay. So let's begin with making a list of those things that accomplish these two jobs, these two goals. So the amount of force a muscle produces depends on the following four things. The following four factors. Okay. Four factors. One, frequency of stimulation of the muscle fiber, read cell, muscle fiber, by a motor neuron. That's one aspect. It's one of the factors. Second is the length of a muscle fiber at the start of a contraction. We're going to go through each of these in some detail to understand what they mean. We're just getting down the ideas first. That's number two. The third factor is the number of motor units activated within a muscle. The number of motor units activated within a muscle. That's the third factor. And the fourth factor is going to be the structural components of the muscle cells being activated. So 4 different factors: frequency of stimulation, length of a muscle fiber at the start of contraction, the number of motor units that are being activated, and the structural components of the muscle cells that are being activated. So let's begin with the frequency factor and see what that is all about. So we'll start with frequency and then move on to the others. So frequency. All right, to understand this idea of frequency, or this factor of frequency, we're going to break it down and look at some beginning initial sort of stimuli, and then we'll put it together into a larger picture. So frequency, the first thing we want to talk about is something that's called a twitch contraction. Now, that twitch contraction is not something that you do when you lift a heavy box or even if you pick up a pen. But it's something that we need to understand in order to understand the larger picture of how frequency affects the force that's being applied in the muscle. So what is what is a twitch contraction? And this is going to be defined by a stimulation of frequency, stimulation frequency, I should put down, of 1. A single stimulation of a muscle cell. Okay. So to put it in words, a twitch is a brief contraction of all the muscle fibers in a motor unit, okay, of a muscle. It is the response to a single action potential from the motor neuron. So the motor neuron sings one single action potential, one beep stimulation of the muscle. So what happens if that were the case? What if we take a motor unit and send in a single stimulation, a frequency of 1? What is the response of the muscle cells? And to understand that, we will look at a graph. So let's go ahead and do that. This graph gives you the response of a twitch, a single frequency stimulation. So along the bottom is time in milliseconds, and a millisecond is 1/10,000th. That should be 1/10,000th, not 1/1000th of a second. Okay. No, that is 1/1000th of a second. Sorry, not 1 in 10,000, but 1 in 1,000. Okay, so a millisecond is 1 in 1,000, 1/1000th of a second. And our stimulation begins here. That's where that frequency happened. Boom, we stimulate one action potential right here. And then we set the timer. And then we chart the response of a muscle. And the way this is typically done is we connect a muscle to a technology that can measure the contraction, the force that's being applied, and plot it out in a fashion like you see this. So what happens initially is nothing. We get this latent period. And then we get a climb. So we're getting force. And then we reach a plateau. And then we get relaxation. So a couple things. First of all, nothing happens initially. And then we get a gradual climb in strength. And then a gradual relaxation. So we don't get this spike. We get a hill. And initially, as I said, nothing happens. So that's called the latent period. Then we have the contraction period, and then we have the relaxation period, and then we're back to state zero. Okay? So how do we explain this? What's going on? Well, let's do that. Let's talk about that. So the machine that we're hooking up this thing to is called a myogram. So we're going to use a myogram. Which is going to be a record of a muscle contraction. And we're going to get this period. And let's go ahead and use this. So this is you should have this. It's one of the handouts. So if you don't have it, go ahead and grab it now. And we're going to mark it up and label it and talk about what's happening at each of these periods. So I'll give you a moment to do that. Put me on hold. And then we're back. So I get this initial period right here. And let me get something that shows up a little bit brighter there. That period right there. Okay. And that's going to be our latent period. We're going to call that the latent period. No force is happening. There seems to be no movement in this period initially. That latent period is around 2 milliseconds. Okay. And what's happening during this period, so during the latent period, we're going to get calcium is being released. Okay. And filaments, the filaments will start to move. And take up slack. So there's a certain amount of slack in this system that we've been describing. It's not taunt. It's not going to react right away. 2 milliseconds is a very, very short period of time. Okay? But it is a period of time. And it's a period in time which calcium has to move from the sarcoplasmic reticulum into the myofibrils and bind to troponin, and tropomyosin has to roll off, and then we're going to connect the cross bridges, and we'll start moving, but we'll take up the slack first. So all of that takes time, and that is represented in my latent period. Okay. And then the next period that you see here is this period. And that is the contraction period. And the contraction period can last anywhere from 10 to 100 milliseconds. Okay. And what's happening during that period is we're getting cross bridges formed, we're moving, sliding the filaments, we're applying force, and the sarcomere is shortening, the myofibrils is shortening, and we're getting this period of contraction, force being applied. Okay. So we dump in calcium. We expose the binding sites on actin. We form the cross bridges. Myosin heads start ratcheting and sliding the filaments, and we're starting to apply that force. But remember, this is a single stimulation, so it's not going to last. So we dump the calcium, we get the movement, and then almost immediately we're going to start taking the calcium back out because that action potential is stopped. We're no longer stimulating that cell. And so now we're going to start reabsorbing the calcium back into the sarcoplasmic reticulum. And that gives us our next phase, which is this next phase was the contraction period. So let's do that in blue. So that is my relaxation period. So here calcium is being removed. Fewer and fewer cross bridges are formed, and we get a gradual reduction in the amount of force being applied. So here, more and more cross bridges, more and more force. We reach an apex. Calcium is now starting to be recovered and removed. So now there is a diminishing in the number of cross bridges, and we get a gradual sliding off of the amount of force. Okay. So that's a single, and by the way, this is going to be around 10 to 100 milliseconds in length. Okay. So that's a single reaction, a single twitch. What happens if we send two, boom, boom, two action potentials, one after the other? Well, it depends. It depends on how fast those two stimuli are received. What is the time period between each of them? Because there is a period in which that cell will not respond to a second stimulation. So within a certain amount of time, if you send a second stimulation, there is no response. After that period, there will be a response. So let's deal first with this non-response period of time, and it has a name. And the name of that is going to be the refractory period. Okay. So, the refractory period. So this is the time in which a second stimulus has no effect on a muscle cell. Now, there's good reason why we have a refractory period. I mean, there is, and you'll learn about that in 40B when you do the nervous system, why that's built into the system. Right now, we just have to understand that it's there. We don't have to understand why it's there or how it becomes this period in which nothing can happen. So if we get a second stimulation within this period of time that we call the refractory period, there is no effect on the muscle cell. Excuse me. So for skeletal muscle, the refractory period is 5 milliseconds. For cardiac muscle, it's 300 milliseconds. So let's talk about that a little bit and try to understand why there's a difference between skeletal muscle and cardiac muscle. First of all, let's understand our refractory periods. So let's say if we are going to graph this, I get my first stimulation here. And I'm going to get the latent period, I'm going to get this, and I'm going to get that, right? Now, that I'm going to get a second stimulation. And I'm going to say this is skeletal muscle, so I'm going to say it's 3 milliseconds later. Again, nothing happens. It's the same graph. There's no change in the graph. For skeletal muscle, that time limit is 5 milliseconds. So anything between 0 and 5 milliseconds, nothing happens. After 5 milliseconds, we will get a different type of response, which we'll talk about in a moment. With cardiac muscle tissue, it's 300 milliseconds, a much longer period of time. Why the difference? Well, consider what cardiac muscle tissue is actually doing. It's pumping blood. And each contraction is forcing blood out of the heart. Now, if you think about that, it makes sense that there is a period of time, no matter how fast the heart is beating, in which blood actually has time to flow into the chamber so they can be pumped out. If each contraction is happening so rapidly in the heart that no blood can fill the heart, then the heart is simply beating with not moving any kind of blood. So the 300 millisecond is a built-in safety factor. So as heart rate increases, if it reaches a certain maximum, you still have a 300 millisecond period of time in which that blood can flow into the heart before it starts contracting a second time. Okay. With skeletal muscle, you don't need that. You need to react. You need to jump out of the way of that train right now. So you want to be able to have a very quick response in between or very short refractory period so you can get that second stimulation when you need it. Okay. So with cardiac muscle, you need a longer refractory period because of the nature of what it's doing. Skeletal muscle, you need a shorter refractory period because, again, of the nature of what it needs to do. Okay. All right, so what happens if we get a second stimulation outside of the refractory period? And let's take a look at this chart and see what happens. And let's get rid of that. So here's our single twitch. We've graphed it. Now here's a wave summation. We get a second stimulation outside of the refractory period, so we get a different reaction. So here's our initial one, and then notice it kind of drops off here, and then it climbs. But look at the stimulation is the same. There's no difference in the strength of stimulation, but we get a difference in the amount of force that's being applied. So with the second stimulation, we get more force than we did with the first stimulation, even though each stimulation is equal to each other. We can carry that out further. I can get what is called unfused tetanus, in which I get boom, 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 stimulation. Each stimulation being exactly the same in intensity, but with each stimulation, I get a gradual increase in the amount of force that's being applied by the muscle. So two things here. I'm getting an increase in the amount of force that I'm generating, which with each stimulation. And if the stimulation is far enough between, there's enough time in between, I get a slow diminishing of force and then an increase, diminishing of force, increase, diminishing of, and then an increase. Let's begin with the explaining why I get an increase in force and why this pattern is not what we want. We want this pattern, a nice, smooth increase in force. This is going to produce a jerky movement because I'm going to apply force and I relax a little bit. I apply force and I relax a little bit. I apply. It's going to be a jerky movement. I don't want a jerky movement. I want a smooth movement. So we'll deal with this after we talk about why we get an increase in force with each additional stimulation. And once again, I'm going to emphasize each stimulation is equal. There is no difference in the stimulation. It's not that I stimulate the muscle harder. I'm stimulating the same way, but I'm getting an increase in force. So what's happening there? Okay, let's talk about that. So two stimuli. And the second one outside of the refractory period. I'm going to get a graph that looks like this. Okay. So I get two stimuli, and I get this kind of graph. So here's my first stimulus. There's the effect of the second. So I'm getting this increase in force with the second stimulus. Why? Well, a couple of factors here are going on. So what's going to happen is you're going to get the following things. So the question is why? And the answer is you're going to get increase amounts of calcium in the system. So with each stimulation, I'm releasing more calcium. With the increase in calcium, I'm getting more cross bridges. Right. Number two, I'm going to get a rise in temperature. Remember, these are chemical reactions that are taking place. I'm splitting ATP and I'm releasing energy in that process. Some of that energy is going to be, a lot of it actually is going to be in the form of heat. And so the muscle cells begin to heat up. And as they heat up, the viscosity of the inside of the cell changes. It's going to become less viscous. Things are going to be able to move more quickly. Okay, so I get a rise in temperature. So that is going to change viscosity. Things are now moving more fast. A rise in temperature will also increase chemical reactions, the rate of chemical reactions. Okay. And there will be also a change in pH, which we don't have to fully understand, but that will also, there'll be actually a decrease in pH, become more acidic, and that will also add the exact mechanism we don't have to understand, just know that it does. So for these three reasons, we're going to get a stronger stimulus or a stronger amount of force being applied with that second stimulus. All right. So that explains why we're getting increased in force. But let's go back to our other problem, which is that in unfused tetanus, which you're seeing here, so-called stair-step effect, you're getting in between the rise a small decrease in force and then another increase and then a small. So that's going to produce a very jerky movement. You don't want that to be your muscles applying force and then relaxing, applying force and relaxing, applying force. You want a nice smooth increase in force with no jerkiness involved. And that's going to be what we call fused tetanus. So fused tetanus is the idea that you're delivering the stimulus at the exact proper rate that prevents the decrease in force from happening. So each additional stimulus is happening right exactly at the time when it prevents that decrease in the amount of force. So there's no relaxation. It's just a constant increase in force. And that's what we call fused tetanus. And that's what your muscles will do automatically. You don't have to think about this. You never thought about this in your life probably until this discussion right now. Your brain is doing this for you. It knows through experience, through when you were growing up as a little baby and learning how to use your muscles, how to send out the right frequency of stimulation to produce a nice, smooth, sustained contraction. So fused tetanus, then, fused tetanus produces a long, sustained, smooth increase in force with no relaxation period between each stimulus. The stimuli are delivered at the right frequency to prevent relaxation. So that's the first factor, frequency of stimulation. And what your body does, and it does so automatically, is it's going to deliver the frequency in just the right time sequence to produce a long, sustained, smooth application of force by that muscle. The second factor is length of muscle fibers on contraction. So this is number 2. And this is the length of a muscle fiber at contraction. Okay. So this is going to describe the relationship between strength of contraction and the position of thick and thin filaments. Okay. So this describes the relationship between the strength of a contraction and the amount of overlap between thick and thin filaments. What is that overlap? That's the question we're going to ask ourselves. And how does that affect the strength of contraction? Well, let's take a look at this chart here, and this will answer our question for us. What we want is optimal overlap of thick and thin filaments. That sounds right. What does it mean? So we're looking for optimal overlap of thick and thin filaments. Let's see exactly what does that mean. So if we look at this chart, along here is force or tension. And along this bottom axis is the amount of overlap between thick and thin filaments. Let's take a look at the extremes first, and then we'll look at the optimal range and talk about that. So here, this is probably the easiest one to understand. Here, there is no overlap between thick and thin filaments. If there's no overlap, then there can be no cross bridges. If there's no cross bridges, there can be no force applied. So if we stretch the muscle enough so that there is absolutely no overlap between thick and thin filament, then we're going to get zero amount of force being applied when we stimulate that muscle. Fortunately for you, your skeletal system will prevent this from happening. So your muscles are arranged on the skeletal system. And the skeletal system has built-in locks. You cannot bend your elbow to the extreme where you're stretching the biceps brachii to the point where there's no overlap unless you break your ankle, your elbow, and bend your arm all the way back at the elbow. That's not going to happen unless this extreme violence has happened to you. So you're not going, this is the extreme case, it's not going to happen. But if you can see that if I just have a very minimal amount of overlap, then my amount of force is not what I would like. It's not optimal. On the other side of the coin, as the filaments start sliding towards the center of the sarcomere, they will begin to overlap one another. As the thin filaments overlap, you're going to prevent cross bridges from being formed. So basically you're eliminating, wherever overlap has happened, you're eliminating the ability of cross bridges to happen, and therefore you're going to lose strength. So there's my two extremes. No overlap, or so much overlap that the thin filaments are sliding past each other, riding past each other, and blocking the ability to form cross bridges. So your optimal position would be where the maximum amount of cross bridges can be formed. That's the optimal. And it's somewhere between 80 and 120. So this is percentage of so this is resting sarcomere length. So that's 100 would be where percentage of optimum. So this is 100% optimum, and then this would be 80% less than optimum and 100% over. So you're starting to reach here. So the optimum is in this range right here, where you're getting 100% over 100% of the cross bridges available being formed. Okay, so that's how the position of the sarcomere or the muscle determines the amount of strength of the contraction. So if you've played any kind of sports, you may have heard your coaches or people tell you to get in your ready position. I know when I was teaching my kids softball, my daughters played softball, and we would, it was always emphasized to get in your ready position, which is in that crouching position, and your arms kind of bent forward, your legs and your knees slightly bent. And what that position does is it places the muscles here in this optimal range. So when you're reacting with all the possible cross bridges that you can engaged in the muscle at the time you want to use that muscle. Okay. So that's the story on the on the overlap, and let's just write down a statement here. Say, a muscle fiber develops its greatest force when there is optimal overlap between thick and thin filaments. Okay. When you have the maximum number of cross bridges formed. All right. So that's the story on the position of the muscle fiber upon contraction. The third factor is number of motor units. So number of motor units stimulated within a muscle during contraction. Do you remember what a motor unit is? Let's talk about that. A motor unit consists of a motor neuron and all of the muscle fibers it stimulates. And muscles are composed of many different motor units. So in this situation that I'm drawing, or that you're seeing in this drawing, I have one motor unit that controls 3 cells and one motor unit that controls 2. If I have a job that doesn't require a lot of strength, then I can just activate a single motor unit. If I have a job that requires a little bit more, I will just activate the green one because I get 3 muscle fibers acting. If I need even more force, I will activate both of them. So this is called motor unit activation or recruitment. Okay. So the number of motor units recruited to do a job determines the amount of force, or is one of the factors that determines the amount of force being applied on a particular contraction. So recruitment is the process of increasing the number of active motor units. Another term that might make sense to you is multiple motor unit summation. The more motor units activated, the greater the amount of force. Okay. Very simple idea. The more motor units you activate, the stronger the force of contraction. At this point, I also want to talk about something else called muscle tone or tension. So this is not directly related to the amount of force that a muscle tension can develop, but it is in that realm. So it's a good time to talk about it. So let's define muscle tone first. Muscle tone is the firmness in a relaxed muscle. And that's why I say it's not directly related to the subject we've been talking about, because we're not applying force. This is a relaxed muscle. But I don't know where else to stick this discussion. I think it makes sense to stick it here because we're talking about recruitment and motor units. So anyway, be that as it may, muscle tone, the firmness in a relaxed muscle. Okay? And this is going to be the result of a small number of involuntary or involuntarily activated motor units in a muscle. So in other words, your muscles are never fully relaxed. There's always some tension in the muscle, and that's the result of some mild stimulation of some motor units within that muscle. Not enough to create movement, but enough to give the muscle some firmness and tension. Okay? This is also important in maintaining body posture. So there is if you're holding your head up during the day, that is because within the muscles that are responsible for holding your head, even though you're not moving your head, it's maintaining its position because there's a certain amount of tension in the muscles that allow that head to stay upright. And that's because we are activating a small number of motor units within those muscles. Okay. Well, the fourth and final factor in producing tension or force in a muscle is based on the structural components of the muscle cells that are being activated. So let's talk about different types of muscle cells. So this would be number 4, the structural components of muscle cells in the contraction or being used in the contraction, however way you want to make it. So it turns out there are 3 types of skeletal muscle fibers or cells. Number 1, type 1, also known as slow oxidative fibers. Okay. These are characterized as slow twitch or fatigue resistant fibers. And they contain the following components. Number 1, large amounts of myoglobin. So we've got a lot of oxygen, high concentration of oxygen in these muscles. Number 2, many mitochondria. So we can produce a lot of ATP. And number 3, high density of capillaries. Surrounding these muscle cells. Okay, so I've got plenty of oxygen. I've got a supply of new oxygen and new nutrients, and I got a lot of mitochondria, so I can produce a lot of ATP with these ingredients here. Okay, so slow oxidative fibers, slow twitch. So even though I have large amounts of the ingredients capable of producing ATP and the ability to produce that ATP, I'm going to use it up slowly. I'm going to lose it. I'm going to use the ATP at a slower rate than in other types of muscle tissue. Okay. So these muscles, they look red. This is your red meat, by the way, when you're in chicken, for example. They have a small diameter. What does that mean, a small diameter? Well, it means there's not a lot of myofibrils in it, right? There is not a lot of these bundles of proteins that we call myofibrils in that muscle cell. So they can't produce very strong contraction. But because of all of this, what they can do is they can generate a high capacity to generate ATP by the aerobic system. So they have a high capacity to generate ATP using oxygen. Okay. And they split ATP slowly. They don't use it up. So they have a slow contraction speed. When you add up all of this together, what you get is a fatigue-resistant fiber. So very resistant to fatigue, but not a strong contraction. These are the kinds of things, these are the types of muscle tissues that are good for posture control. So postural muscles are made up mostly of these types of muscle fibers. They would also be nice to have if you're running a marathon. The second type, number 2 here, is, oddly enough, called type 2A, and this is fast oxidative fibers, okay, or fast twitch. A fibers. These have a medium diameter, so they produce slightly stronger contractions, and they contain lots of myoglobin. So we've got lots of O2. Lots of mitochondria. So we can produce a lot of ATP. High density of capillaries. So we can resupply these muscles very, very efficiently. They can generate a lot of ATP for the reasons we've just listed. Okay. But they split it fast. Very fast. So they have a very fast contraction speed. So they can resist fatigue, but not as well as type 1. And this is the kind of muscle that you might want in a sprinter. So running a fairly fast, short distance. Because you're going to be able to generate a lot of energy, apply a fair amount of force for a short period of time. Okay. The third and final type of fiber is the type 2B, or fast glycolytic fibers. Notice that we've changed the term here, glycolytic. Okay, so here, the fast glycolytic, this is referring to a way of generating ATP without a lot of oxygen. The first two types of fibers that we talked about were capable of generating ATP using oxygen. These muscles are more in tune to generating ATP without a lot of oxygen present. So they are fast twitch. Okay. They are the thickest. So they're very powerful muscles. And the content is we have low amounts of myoglobin. We have low amounts of mitochondria. We have low density of capillaries. But, number 4, this is the kicker, we have large amounts of glycogen. So we got plenty of fuel, but we don't have the capacity to generate ATP using oxygen very well, because there's not a lot of oxygen available in the cells, stored in the cells. We don't have a lot of mitochondria, and because we have a low density of capillaries, the delivery of oxygen is going to be impaired. However, we've got huge amounts of fuel to burn, and so we're going to be able to burn that fuel using the glycolytic metabolic pathways. So what we get is these are good for endurance types of exercise. or we can get, and that's not endurance, but short, strong application of force. Throwing the ball, lifting weights, this kind of thing. A couple of points. Exercise can change or produce changes in muscle fibers. So you're not set with a certain type of muscle fiber all your life. That can change over time. So for example, endurance exercise can change a type 2 to type 1. Strength exercise can increase the number of type 2B fibers. So these are the 4 mechanisms which can generate force within a muscle and which can allow you to apply different amounts of force to a particular job. Okay. So ladies and gentlemen, that's the end of this lecture. And in the next lecture, I'm going to deal with metabolism and energy sources for muscle tissue.